random shearing by physical or the enzymatic means, we can generate millions of reads. So these reads may be in the paired end oriented form, or they may be in the single end form, right? So either they will be single end or the paired end, depending on the chemistry and then the method we have used for the sequencing. Once these reads are there, we can form by aligning these reads to form the longer contigs here to get that template DNA which we have sequenced, the genomic DNA, right? So to assemble that into longer sequences, and those sequences which are assembled there, they are called the contigs. Right. So this assembly process, we cannot do it manually because these reads are too many, like millions of reads are there. And if we start aligning them manually, it will take a lot of time and that will create various errors in the joining of these reads or aligning these reads. So there are different assembler softwares available at this system for the assembly of these sequences. Once we have the contigs here, we have the paired end reads with us. So we can try to join these gaps here. Suppose there are many different contigs generated. So what we can do, we can use these paired end data here, paired end reads, forward and reverse reads to align them and then finding the two mates to mate contigs there and creating the longer sequences. They are called the scaffolds. So these scaffolds may have the filled nucleotides or they may have the gaps in between. They will be represented by the ends there, right? So this is the whole assembly process here, how it is being performed. I explained it to you yesterday about the paired end sequencing here. Okay, please clarify me if I'm not audible to you. Am I not audible to you?
Sir, you are now audible, but few minutes ago you wasn't
I can
audible.
switch off my video. That will improve the network quality also. So let me stop the video first. So that will create much better network. Right. So I hope it will make it better. Right. So paired end sequencing, when we do the sequencing from both the ends of the template DNA, what we have applied on the next generation platform, next generation sequencing platform. So forward lead and the reverse lead. Same template DNA sequence from both the ends. Two ends are used to sequence it for the same template DNA. Utility of these paired end sequences are like they are good for the repeat region assembly there. So we can assemble the repeat regions and the software has the benefit over the single end sequences to assemble the repeat regions there in which are present in most of the eukaryotic genomes and also in the prokaryotes as well. The second advantage, I told you that we can also create the scaffolds for longer sequences. So we can still fill the gaps between the contigs if they are present. So we can use that information using the paired end data, or the mate pairs are used for the longer stretch of the DNA. Right. So those mate pairs can also be generated by using the longer sequences. So you can read it much about the mate pair and then the paired end inward outward sequencing. So those are the mate pairs and pair reads are used to assemble the sequences and filling the gaps there if the gaps are present, right? So this is the general workflow of the genome assembly and annotation. So here suppose we have the raw FASTQ sequences what we saw yesterday we had, and then we can use these FASTQ sequences to get the high quality FASTQ reads here. These high quality reads can be generated by using the FastQC toolkit, like we can get the quality. Then there are different tools like trimmers also available, like PrintSeek, NGS toolkit, Trimomatic, Cut Adapt, Adapter Removal, and various other tools are also available. So we can use any of those tools to process the data here to get the high quality. Once the data raw sequences are of high quality, we can either follow the workflow of the assembly and then the genome annotation and comparative genomics based on the draft genome or the complete genomes, whatever we receive after these steps. Right. The other way we can find here, like we can use these HQ FastQ sequences high quality sequences to align and then to the reference genomes, and then we can use that alignment for the variant calling, SNPs, prediction, and comparative genomics again. Right. So today we will be working at this step, assembling the genomes into context scaffolds or the circular. So depending on the quality and the amount of data we have, we can get the draft genome or the complete genome. We will use here the genome of a prokaryote, and the tool which can be used for the assembly process, they are like Spades, Velvet, CLC Workbench, which is a paid software. So it is not utilized here. We can use either Spades, Velvet, SoapDenovo, IDBA, Canu, etc. So these assemblers can be used. MegaHit also there, so you can use those assemblers there. Then tomorrow we will work on the annotation of the draft genome or the complete genome. So you using different tools like Proca, Rast, CAR, PGAP, and CAR, anti-smash database, you can use them. So what we will focus mainly on the Proca, Rast, and CAR server annotation. Okay, so we have the template DNA or the any genomic DNA we have there, we have sequenced it. So how we can identify the coverage? Like coverage is very important because we want to know it's the high throughput sequencing platform, and we don't know that how much coverage we have received from the platform there, like after the sequencing. So the coverage tells us that how many sequences are representing that particular template DNA which we have sequenced there, right? Or genomic DNA which we sequenced. So length of genomic segment here, suppose it is denoted by the L, and number of reads is N. So how many reads are being generated here, and length of each read. So how many base pairs are there for each read. That's how we can calculate the coverage of the sequencing data. So suppose here the coverage is N into L. So that will be the number of reads and then the genome multiplied by length of each read. Then divided by the length of genomic segment, right? So suppose we have here 1000 base pair of the genome which we have sequenced. 200 reads were produced, for example, and each read has a length of 51 base pairs. So coverage will be 200 multiplied 51 base pairs divide 1000. So total sequencing coverage will be 10.2x, right? For Illumina data, what we receive from the Illumina platforms, we need to have at least 60, 70 or 100x coverage. More is better, but less is not good for the downstream analysis. So it is always better to have at least 100x coverage or 80x coverage, more than that. So when we do the assembly, there are two methods can be used, or different algorithms are also available. Depending on those algorithms, different tools are available. So one method is the alignment method, where the reads are aligned one to one, and then we receive the longer sequences there. But that has the limitation of utilizing lot of memory, like primary memory, and then the computational resources like CPUs also. So to overcome that, there are different other algorithms were also developed. One of them which is based on the k-mer based assembly. So what are the k-mers here? Suppose we have this initial segment of DNA or genomic DNA here. We can sequence it and receive the various different reads. Right. These reads can be used to align and then get the sequences. But what we can do using the k-mer based approach here, what we can create all the possible nucleotide mers. So these mers can be different short stretches of the DNA by using the creating here, like suppose in this example it is 4-mer, but in real life when we use the k-mer based assembly, most of the tools they use odd number of the k-mers, not the even number, like 4 is repeated here. But in this example, I'm showing you for the even number, right? So how the k-mers are generated, suppose this is the ACTC. So the first 4-mer will be generated using ACTC, and then one slide window up here, and then that will be the next 4-mer will be CTCG, and then next will be TCGA. So all the possible 4-mers will be created for the reads here. Once these 4-mers are created, then exactly or the same formers will be discarded from there, and then there will be alignment of k minus 1 alignment for these formers. After that, the consensus sequence will be created at the by finding the lowest path there. So finding the De Bruijn graph, creating the De Bruijn graph. So Velvet and Spades, they use the De Bruijn graph constructions. If you are more interested in that, that is a mathematical model. So you can go and read about those methods on the algorithms which are available. But the here which is used like Kramer, and then following the creating the De Bruijn graph and following the shortest path to create those consensus sequences, finding out them. The reason why the odd number of the k-mers are used, not the even number, like 4-mers are not used in the assembly. So when we select the k-mers, they are odd number, like 3, 5, 7, 9, 11. Why? Because they avoid to form the palindromes in the sequences. If we use the even number of the k-mers like 4-mers, 6-mers, 8-mers, they will form the palindromes, and then that will confuse the assembler that which way is to follow there, and that will create the errors during the assembly. To avoid those errors, odd number of the k-mers are being used, right? So one assembler here, that is Velvet assembler, which is based on the k-mer based assembly and followed by the deconstruction of De Bruijn graph. It has two modules, Velvet H and Velvet G, right? So you have to first run the Velvet H to create the hashes of the data, and then it will give the input, it will create the input for the Velvet G there. Then this Velvet G will take the input from the out of this Velvet H, and then it will build the De Bruijn graph from the k-mers which were generated here by the Velvet H, and then create the longer sequences, contigs, right? Another assembler, that is Spades assembler, which was developed for the assembly of single cell genomes, right? Why? Because single cell genome assembly is a little bit tricky process. They are during the sequencing of single cell genome, there is multiple displacement amplification, and that generates uneven coverage of the sequence outputs. So to overcome that problem during the assembly process, because these assemblers, they rely on the coverage also of the reads. So they have modified that algorithm to assemble the single cell genomes. But now they have also incorporated the assembly of isolates, right? And then the metagenomes, transcriptomes, RNA-seq assembly or has also been added there. So it has different steps there, like error correction, De Bruijn graph processing, repeat resolution, like resolving the repeats which are present there. Then the post processing of the data, finally contigs are generated in this process. So this also works on the k-mer approach, and then the formation of De Bruijn graphs, and then the assembly process. It also does the error correction using its, I think there is a tool, I forget the name, I will tell you if I remember that. So it uses the error correction within the reads. So it uses different k-mer values, and then looks for the errors if it finds, and corrects those cases, and then you use that data, corrected data, for the assembly process, right? So if you have questions, you can ask till now here. Otherwise, we can move to the Spades assembly, and then we will also check the quality of the assembled sequences, so contig sequences using the Quast. So we will use here two tools today: Spades for the assembly of high quality short reads, and then the quality control analysis of the assembled contigs. So how we can compare or analyze the assembled sequences, whether they are of good quality assembly or bad quality assembly, right? So do you have any queries, questions?
No, yeah.
No. Okay. Okay, so what is not clear to you, Hadia Khan? What is not clear? Can you tell me, like this one? Okay, you are asking about this one. Maybe. Yeah. So what is not clear?
Sir, I have one small doubt.
Yes, please.
Uh, Sir, while calculating the genome coverage, we have to use both the reads pair, means read one and another paired and also or only one read.
Both the reads and pairs. Yeah, read pairs.
Okay, sir. Thank you.
So you can do the coverage analysis using the SAM tools, which is available there. So you can align those reads to the reference genome. And then there is the function and which is built-in function for the SAM tools. You can look for the coverage of this from the SAM file or the BAM file. You can calculate the coverage. There are different tools also available. Other tools also available for the coverage analysis. Okay, how they are created. So the creation is here. Suppose we have these reads which are generated from the sequencing platform, and these reads are used for the uh, formation of or creation of the all possible k-mers, right? So k-mers we can define here, like suppose we want 5 mers, not the even number, odd numbers are used. So we can define like 5 mers we want to use. So those 5 mers will be taken from this read particularly, A, C, T, C, G. That will be the first 5 mer. Second 5 mer will be created C, T, C, G, A. And then the third 5 mer will be created C, T, C, G, A, T. Right. So that these are the all possible 5 mers which will be created for all the reads here. After the creation of these 5 mers, all the duplicated or the similar 5 mers will be discarded from here and they will be aligned here as the k minus 1. These ones. So this is the alignment which is shown, but the tools like Velvet and Spades, they use the De Bruijn graph. So they align these ones using k minus 1 approach and then travels those shortest paths there using the De Bruijn graphs there which was constructed, right? And then the consensus sequences are deduced, right? Why we take in odd no forms? Can you explain it again? So odd numbers why we select, because they cannot or they will not form the palindromes there in the sequences. Odd numbers, there will be one extra nucleotide that will disrupt the formation of palindromes, right? And if we use the even number, those k-mers will form the palindromes, and that will create the errors during the assembly process and assembly outputs. That's why odd number of the k-mers are used, not the even number. Okay, so this Spades tool will be used here for the assembly. Here you can see these two examples. We can run it at default parameter. So I have run this one at default parameter here, which we see, and we can also define the k-mers here. So these are the randomly defined k-mers like 31, 45, 55, 75, 85, 95. Like these k-mers are randomly defined here. Right. So let me go and show you how you can install the Spades and use it. Website of, yes. Here is the official website of Spades. right? You can get the manual of Spades here. Spades manual with installation guide, version 3.15.4. Right, so this you can get here, the manual of Spades. Then you can also find the GitHub page. Look for a simulation GitHub repository. You can go to the GitHub page and then you can download the recent version, or you can also download the binary of the Spades here. So download Spades binaries for Linux 64-bit here. So you click on it. It will download that particular Spades tar.gz binary file. Then you can uncompress this file by putting it somewhere in your PC. So I'm not going to save it because I have already downloaded it. I will show you once you download it. So you can download it like here, where you want to do, like go to the drive and then go to the particular drive and then save it there. Right. Once it is downloaded, then you can go to the terminal. Right. So you can go to the MNT D, CD, MNT D, and then the drive. So here are all the data which I have processed yesterday for the high quality. Those paired, unpaired reads from the Trimomatic run. Right. So those reads are kept here in this directory. And this is the Spades 315.4, 3.15.4. This version you can download in this directory in the D drive, right? Or C drive or anywhere where you want to download it. And you can uncompress it. This is Spades tar minus ZXVF and then the tar.gz file. So this will be Spades tar.gz. You can press enter. It will uncompress the file and create a directory name Spades 3.15.4. Here you can see Spades 3.15.4. If you check here inside this Spades directory, so I'm going to see here Spades because it's a binary output or binary file to we can run it directly on the Linux platform. So we can go into the bin directory and there is spades.py. Right, we can run it directly. So the one way to use Spades is this one. The other way is to install it using sudo apt install Spades. Right. So other way to install Spades is this way. This will install the stable version of the Spades, like the older version. This is the recent version, which is not the stable one, right? So you will not get this version, but you will get the Spades 3.13 point something. And the third way to install the Spades here, if you have the Anaconda or Mini Conda, then you can use here Conda install -c Bioconda and then Spades. You can download this using the Mini Conda also or the Bioconda. You can install it. Right. So these are the three methods you can use this Spades. Once the Spades is done, this directory is there. So suppose you want to keep it in your software directory. You can keep it there and then you can export the path to your bash RC file. Right. So that will add it to your system path path there. Like this Spades installed as the spades.py is installed as the sudo apt install, which is the older version. Right. This is the version 3.13.1. Right. So you can install any of these versions. Depending on those versions, you can use the different options which are available. right? So once the space is installed, you can check it using space.py -h. This assembly I have performed today at the default parameters. So the next assembly we will do here using the defined Kmer parameters. So I have used the space which is in the user lib space bin. Okay. We'll use the
Hello, sir. Sir, after unzip my file, which code should I run for install this application?
how you can run this one. You can, whether you have uncompressed it, okay? Once you have uncompressed your file, you can check that uncompressed directory. Like here, you can see like this one space. So you can type the command this one and then the uncompressed directory name space. Then there will be bin binary directory which will contain all the binaries. Then there will be space.py, right? You can run it like this. Okay.
Yes, sir. Okay, I understood.
Okay, so space.py. Right. We can check the help of the space here. What are the options available? So as I told you, all the output will be created in the provided output O option. So we can provide the output directory name and all the results will be created in that directory. Yes. Then the SC option is used for the single cell. SC is for the single cell. Meta is for the metagenomic assembly. RNA is used for the RNA-seq data assembly. Plasmid option for the detection of plasmids and extracting those sequences. Ion torrent for the ion torrent data sequences. If we have the ion torrent platform data, we can use this flag also, ion torrent. Test will run the space on the test data set to check whether the space is working fine or not. Right. I showed you like -h and minus help can be used to see the options and then the uses of the different flags here. The input data can be given here as the -1 2. So interlaced reads can be used or the forward and reverse reads separately can be provided. So what is interlaced data or the paired and data? That is like first 4 sequences will be the forward, then the next 4 will be the reverse reads. Then next 4 will be forward and next after that 4 will be for the reverse. So that is the interlaced data. -1 for the forward paired and reads and -2 for the reverse paired and reads. Minus n we can -s use it for the unpaired reads also during the assembly. Right. Then if we have the merge sequences, suppose we have merged forward and reverse reads there. So we can use that merged flag here and provide to the space those merged file here. Then the library names can be provided using the different libraries and we can use those library numbers right there and providing those files. Right. Mate pair data can also be used here. Like the pair and data, we can also use the mate pair data, which is obtained from the longer stretch of the sequence DNA template DNA, not the like 300 400 base pair, but maybe it could be like 1 kb 2 kb or 3 kb of the is between the stretch. So that will be used for identifying the contic sequences, whether they are from the same stretch or they are from very far apart. Right. So that mate pair libraries or the sequences we can also provide here. Sanger reads can also be used here. Sanger option and then the file name with the Sanger reads. PacBio nanopore reads can also be used in the space. So we can use that data as well. Trusted contigs, TSLR contigs can be used here also. Like what are the trusted contigs? Suppose you have previous assemblies and you know that this one is the good one and you are doing the reassembly, then you can use those trusted contigs giving there. Untrusted contigs you can also create here. Right? Pipeline options here like only error correction. So it will do the error correction, not the assembly, and only assembly, no error correction. So these options can be used. Careful for the mismatches and the indels will be reduced there. You can run the space at certain checkpoint. So use the continue option and then it will run from that particular checkpoint. Restart from using the different parameters. So you can specify the checkpoint here and then run it again. Right, these are the threads and then the memory options. So you can use how much you have available with you. By default, it uses 250 GBs of RAM with 16 cores. But if you have less, you can use less also, and more you can define here when you are running the speeds. Right. Okay. We can run it. Speeds -1 will be the forward read. So we can use here forward, paired, and read, which is forward pair -2 reverse pair -s unpaired and -s unpaired. Right. -k we can use here. Right. 31, 45, 55, 75, 85, 95, 105, 115, and 127. So the limit is 127 merge. Depending on the read length, if the read length is 100, 100 base pairs, we cannot use the 105, 115, and 127. We can only use till 95. Right. But the reads are up to 230 or 240 base pairs long. After the trimming, we can use 127 of the gamer size. Right, we can also define the -t threads. I'm using 3 because 4 is available with me. 4 threads are available here. Then we can use -o for the output assembly defined gamers. Okay. If you are using the recent version of the space, so you have to provide there, I think the isolate option. But in the 3.134, it is not required. So I will run this. Someone is asking SMRT data also being used. So I haven't used the SMRT data and I don't see the option here, but the long reads can be used, like from the nanopore and then the pack bio also. So I'm not sure about SMRT. I think you can do it. Yes, I found a paper here where they have used the SMRT reads and then assembled the data from the single cell. So I think it is the hybrid space they have used, which is implemented in the space software, space assembler. Okay. So I will share this link with you. Hybrid space, yes. Right. So just look into this paper. So I will suggest you to read more about the like space manual and other tools which we are saying here or we are using. You just go through those tools and their manuals, how you can use those tools there. Right. And what are the options and what kind of data and what kind of the input you can receive when you use certain different options or the flags when you apply them. Right. So it will be much better to understand what the different tools and also their functioning, and also that will also clarify you like which options should you use when you have this type of data or certain type of the output there. Right. So there is not much you can like read by previously, but yes, certainly you can read like Spade's paper and the other assemblers paper. If you are interested in, you can go through those papers and the review articles various available.
Hello, sir.
Yes.
Sir, when FASTQ file is merged, that is pair 1 and pair 2 both are merged, how to separate, sir? How to split them?
Merged means they are interlaced.
Yes, sir. After read 1, there is read 2. Like that, sir. Pair 1,
Okay.
pair 2.
So you want them separately. To
Yes,
do
sir. I
it
want them
separate.
separate. Okay.
There must be some tools available to do that. Otherwise, you can write your own script to select those sequences from the like every fourth sequence, like fifth sequence, should be extracted from there, right? So you can write a Python script, or even the one-liners will work very well on this. What you want the first sequence, and then take that into separate file. So print that. Then the second sequence should be in the other file. So you can write your own script if any tool is not available, but there must be some tools available. I don't know exactly, so I can't suggest anything here about those tools. But you can write the scripts. Yes, I'm not speaking anything yet. So if you have questions, you can ask till it finishes. So I'll try to answer, but it will take like 2, 3 minutes more to finish, I think. Okay, so this is finished. You will see how the data is there in the output directory. Sorry. Here is the two outputs we have. So I ran this one earlier on default parameters. So let's check the output for this default one. Here you can see 21, 33, 55 and 77 kmers were used. But the reads were of length of up to 240 or 230 bases long. By default, Spades uses these 4 kmers here, right? And gave us the output of this scaffolds.pasta if it was able to do the scaffolding, otherwise the contigs. So these scaffolds are supposed to contain the filled sequences. So that could be the nucleotides or they are filled with the ends there if it was able to do the scaffolding, right? This is the contigs. So these contigs will not have the contigs ends there if ends are not available in the sequence data or in the high quality reads. If ends are present there, that might create the ends in the contigs file also. But ends are not, ends will be present in the scaffolds.fasta if the scaffolding is performed, right? So these two outputs we see here. There is the space.log file also available. So you can always come and read what parameters were used and when you ran this assembly with what parameters and all the steps and then the processes are recorded in this log file, right? This is scaffolds.fast and context.fast. If we open this one, you can read here node of the node means number of the sequence. So it is the sequence identifier, node 1. Then length 191,000 base pairs, right? And then coverage is 31.04. So it also calculates the coverage, which is important in the case of assemblers, right? So here is the coverage, length and name. If we go at the end of this file, you can see there are 109 sequences present here, 78 length up to 78. But what sequences we can select here for the prokaryotic genome, that is 200 base pairs. So sequences which are longer than 200 base pairs, they are good to take. We cannot use the sequences or submit the sequences to the public database which are smaller than 200 base pairs. Right, so up to 200 base pairs, it's fine, but less than 200, they will not be accepted. Ends are not accepted also. So scaffolds are not submitted to the public databases. They can't be, we cannot submit the scaffolds. Okay, only the contigs are accepted there. We go in the other assembly output, defined one, where we define the kmers. Here we have starting from 31 till 127. All the kmers are odd number. None of them are even number kmers used here. And then the output files is scaffolds.fasta and contigs.fasta we have here. Initially, we can check here how many sequences are there in contigs. So in the other file, there were 109 sequences at default parameter. We see here, and the 53 or 52 sequences were longer than 200 base pair. Here, if we check the assembly output, we see 27 sequences are longer than 200 base pair. So initially, it looks better assembly here for us because we have less number of the sequences. So it means that sequences are longer. But still, we don't know whether the nucleotides or the amount of this assembly or the sequences are good enough to use or not. So to do that, we can use the QAST here. Conda info QAST. So you can use the QAST tool to check the quality of assembly. Right, so QAST is installed in the Anaconda here. You can install it using the Conda install. So you can create an env like Conda create -n QAST. Then once it is created, you can run the command conda activate QAST. Once it is activated, you can run conda install -t bioconda and then QAST. It will install the QAST in the QAST env, and you can always activate this env and deactivate it. So once it is installed, you can check the QAST here using QAST -h. How you can use the QAST tool. Right, so there are various available options there. You can use. The required ones are output directory is required. You can use the reference genome. If you have the reference genome, you can take that genome for the comparison. If you have the genomic features file like GFF, BED, NCBI or text file, like you have the features for that genome, you can provide that for the reference. Minimum contig length is 500, but you can select a 200 for the prokaryotes, or for eukaryotes you can select 500. Then you can define here what genome you are using. So by default, it doesn't require, but if you are using the eukaryotic genome, fungal or large genomes, so you can define these options like this is eukaryote, fungal genome or the large genome. You can define it. K-mer stats, you can also use a compute K-mer based quality metrics. Right, so these are the options you can explore for the QAST here. So the main one I have showed you there, like we can use the reference genome. Along with that, we can use the minimum contig length. Right. So I will copy those sequences here. I will go out, copy those sequences, contig sequences from the default one as the contigs.faster as the default.faster assembly. And I will copy next one, defined one as contigs.faster as defined.faster. Okay. You can use as many as sequences you have, like you can use different assemblers also, and then compare the assembly, those assemblies here using the QAST tool. To do that, QAST -o will be QAST out, and then -r. So there is one genome which is Mycoplasma pneumoniae strain 11, which may be close to this genome, so we can use this as the reference. And then the input files. So input files are default.faster and then define.faster. Also, we will use here -m option for the 200 because by default it will take the sequences of 500 base pairs. as we did not give the features file, so it is saying no file was with operons and minus o option were given. So if you have those files like gene finding option, you can also use here. So these options are given in the Quast help menu. You can check these options always and then apply them. Okay, so this is finished. The output is created here. You can check the notices for 0 warnings and non-fatal errors 0. So this is finished with no errors. Quast out, we can go inside this directory. There are different files. So you can check it here. Report.html, PDF format output is there. .text, text, TSV, TXT. So these are the different output formats. You can use these formats depending on your requirement, like you want to use it, analyze it. So those results you can use it from here. We can open this HTML one and visualize it in any of the web browser. So I'll go and then share the screen. Okay. Here is the output from the Quast, right? We can see the genome stats based on the genome alignment. There are the default and this is the defined assembly. So we use the defined k-mers here. With respect to the reference genome, this covers 97% of the assembly. This covers 98.8484% of the assembly with respect to the reference genome. Duplication ratio is 1.001. Largest alignment for the defined one. When we use the k-mers starting from 31 to 127, not in the default one. Total aligned length is higher here for the defined. NGA50 means the genomic alignment. So that the sequence which covers the 50% or more of the genome. So if we use 95,000 length or longer than these sequences which are available, that will cover the 50% or more of the genome, reference genome. Here it is lower. So till now, it is saying that this is a good assembly. Number of sequences which are covering the 50% or more of the reference genome, that is 3 here and 4 here. So it is still better now. Now, we can see here in terms of the misassemblies now. This is 2 misassembly, 3 misassemblies. So this shows that there are more misassembled sequences or misassemblies available compared to the reference genome. Misassembled contig length here, which is longer for this. So it says that this is the problematic error. But we are not sure whether this sequence, that this reference genome is correct or not. It is the same one what we are comparing with. If we are doing the resequencing of the same genome, it would be good to compare here. Or we know that this is the most closest genome which we are comparing. Then it's fine. Otherwise, we cannot say that this is a misassembly or this is the correct assembly here. Right. Then we can see here stats without reference genome. So this is the stats without reference. Number of contacts 57, longer than 200 base pair when we put that filter parameter for 200. Here 27 contacts are longer than 200 base pair. So this is obviously better. Rest all of the stats which without the reference genome, which says that this one is the best assembly, defined one, compared to the default one, right? So based on this output or this report, we can select that defined one or defined assembly is better, and we can select it for further. These are few of the stats output in terms of the plots, like the cumulative length of the genome we see here, then NX, like how the genome is covering with the number of different sequences when we add them. NAX is the how the sequences are covering the different percentage, like 39.05%, and then the length of the sequencing, right? It's 62%, and then the different length of the sequences and how much fraction of the genome they are covering. This is for the NGX and NGAX. So this is for the with respect to the genome alignment, reference genome. So how they are covering the different fractions of the genomes here when we align those sequences to the reference. Misassemblies plot here. So what are the sequences? Like 0 misassembly, 0, 1 misassembly here for this one. And then defined one here. Second misassembly, and then the third misassembly here. 97.64% as the coverage. Right. So these are the different plots we received here. This is the GC plot, which is exactly same for the reference and then the genome which we have used for the assembly, which is in the range of 40 to 41%. Right. So this is the few of the example of the assembly and then the quality analysis, how you can do the comparison of different assemblies. So this is not only the single tool and different parameters, you can also use here the different tools and then the different parameters. You can combine them and use them here to compare and select the best assembly based on the stats. So most important one is the L50, N50 are the parameters which are the most important. And if you have the same genome, you can select the misassemblies also as the parameter for the good assembly. All right. So these are the few examples, and then I would like to wrap up the today's session with these ones. And tomorrow we will see how we can use this particular genome here to do the annotations, so using different tools. All right. If you have queries, questions, you can ask. I'll try to answer. Otherwise, we can close the session.
Hello, I
Yes.
have a question, please.
Yes.
I want to ask when we are going to start to talk about variant annotation and variant calling and comparing it with the population database and genome ad.
I think the
Are
GWAS
you going
study. I think um, that has been replaced. I don't know what is the schedule there, but you can ask the organizers. You can write them like whether they are including it or not. Okay.
because in the I registered already in this course
Yes.
because for this session the in the schedule it is already should be today variant calling one
Exactly. Yes. So I
and
have been
variant
I have
calling
been
two.
informed very shortly, like on the short notice. I have been I'm here to present these two modules like today and tomorrow, and also on the 12th and 13th. Okay. So you can ask the organizers. They can organize something for you, or whatever can be done.
But can you tell them because you are one of the people who are involved in this workshop just to tell them that
Sure. Yes, I will talk. Yeah.
yeah, I'm really interested in these two sessions and I
Yes, the
already paid
GWAS
for
study. I
this
think you were
workshop.
interested. Yeah.
Yeah,
GWAS. Yeah.
to Yeah, please. And because there's still another 2 weeks, so they could arrange something for us about this.
Yeah, I will talk here.
Yeah. Okay. Thank you.
Thank you.
Sir, can you give us some project paper that you do as a project, then you publish it in some journal? Can you give us some paper?
Okay, so these are like the basic analysis which you do. Okay, after that you have to do like assembly. So you will find lots of these genome announcement papers, right, which are published in the Genome Announcement Journal, and also you can find it in the comparative genomics literature or the articles. These methods. Okay.